But briefly, Amelia is a professor here at Mississippi State College of Veterinary Medicine. She's been here since 2015. Prior to joining the faculty at Mississippi State, Amelia spent 17 years at the University of Georgia as a faculty member there. She's a DVM received her DVM from Purdue, received her PhD from the University of California, Davis, and worked there under Dr. Laurel Gershwin, who we're familiar with. Amelia has just a ton of things going on in terms of the research, and her focus has always been on respiratory tract disease. And I have to tell you that since she's been here at Mississippi State, she's just been a beehive of activity. She currently has 3 funded USDA projects, which having just one is usually sufficient for most people. She has 3, and they all focus on BRD. And so she's a very busy person. She also participates in our teaching and our clinical program. So I'm going to, I'm going to go ahead and move to Amelia's video. Amelia is online, and we will have Q&A right after her video. So just bear with me for just a moment.
Thanks for tuning in tonight to learn a little bit about the role of inflammation in bovine respiratory disease. So we'll talk first about the genesis of the inflammatory response, and then we'll review infection and inflammation in BRD, and then finally we'll finish up discussing some of the evidence for benefits of anti-inflammatory therapy in BRD from both experimental challenge studies and also some field trials. So this picture is just to remind us that the calf is born into a world populated with millions of microorganisms that have the capacity to enter her body and take it over, causing disease or even death. And so the calf has to be able to defend herself on a variety of surfaces, including her gastrointestinal tract, her respiratory tract, her mammary gland, her genital system, and even her entire body surface against a range of different organisms. She has to also be able to clear the organism if it does enter her body without causing too much harm to herself or to her unborn calf. And so in order to do this, the host has developed both the innate immune response, and that's the component of the immune system that is immediately and always active. And it responds generally to a broad variety of agents. The immune system also has the acquired or adaptive arm. This arm takes several days to weeks to become fully active, and it only responds to a single agent, but it does improve with repeated exposure, and this leads to what we know as immune memory. And this is the component of the immune response that we target with vaccination. Now, I used to say that the innate immune response didn't display memory, but recent research has shown that the innate immune response can actually improve if animals are exposed to stimuli over time. And this has been called immune training, and that's to distinguish it from immune memory. And so you may be hearing more about immune training in the future. Mechanisms important in the initial response to infection are also involved in the response to non-infectious tissue injury, such as occurs with tissue trauma. Both infection and tissue injury cause inflammation, and inflammation activates the immune response. So if we have infection with microorganisms, these microorganisms contain PAMPs, pathogen-associated molecular patterns, that are recognized by the immune response. Now, if you have tissue injury, cells and tissues are made up of molecules, some of which can be recognized, and these have been termed DAMPs, damage-associated molecular patterns. Another term is alarmins. In either case, PAMPs from infectious agents or DAMPs from damaged tissue are recognized by sentinel cells that occur throughout the body, and these sentinel cells have pathogen recognition receptors that allow them to identify PAMPs and DAMPs. When they do, they release molecular signals, cytokines and vasoactive molecules that lead to an influx of innate immune cells and the vascular changes associated with inflammation. And this leads to the infection or the tissue injury being cleared. And sometimes if the infection or tissue injury isn't very severe, the innate immune response can take care of it all by itself. Or if it's more severe, then the acquired immune response may become involved in a continued effort to remove the infection. Now, these pathogen-associated molecular patterns are highly conserved molecules found in many different microorganisms, and that's really a good thing for the host. The host response has evolved to recognize these, and basically they allow a relatively few molecules to initiate immunity to the effectively limitless microbial world. Cells are able to recognize these PAMPs with their pathogen recognition receptors, or PRRs. So a few of the important PAMPs include peptidoglycan, which is a component of the cell wall of gram-positive bacteria, lipopolysaccharide, or endotoxin, which of course is a component of the cell wall of gram-negative bacteria, unmethylated CpG nucleotide motifs. I know that's a mouthful. That's basically DNA that has C's and G's on the same strand that lack methyl groups. And these are commonly found in bacteria and sometimes in certain viruses. And the whole point about unmethylated is that in the host, in the mammalian cell, if you have runs of C's and G's together on the same strand of DNA, they will have methyl groups associated with them. The fact that they are unmethylated is what clues the host immune response into the fact that they likely come from bacteria. And then another common PAMP is double-stranded RNA, which is found in many viruses. Just a few words about damage-associated molecular patterns. Some of them are things that come from extracellular space, such as small fragments of hyaluronic acid or fibronectin or collagen. You may remember these as sort of intracellular matrix components you learned about in histology, or certainly components of tendons and ligaments, collagen, and also the subcutaneous space. Intracellular DAMPs are things like unmethylated CpG DNA from mitochondria. Now that's interesting. Mitochondria, which are inside of cells, were, it seems likely, prehistoric bacteria that at one time then became incorporated into cells, and they've lived happily ever after. But the mitochondria do contain their own DNA, and it happens to be unmethylated, or it happens to contain relatively large amounts of unmethylated CpG motifs. And so what that means is if your cells are damaged so badly that the contents of the mitochondria are released into the extracellular space, that's going to activate an immune response. Adenosine from ATP and nucleic acids is a DAMP, as is uric acid, which is a breakdown product of purines, which of course are also components of DNA and RNA. I just want to go back to this point about small fragments of things like collagen causing an inflammatory response. When collagen is in its regular sort of repeating unit form, it does not at all induce an inflammatory response. It's only when it's broken up into small soluble fragments that it can induce inflammation. So normal healthy collagen is not going to induce inflammation, but it can induce inflammation if it's broken up into small soluble components. And I'm not going to get into this too much more tonight, but you may already be thinking about the immune response activated by DAMPs from injured tissue could lead to autoimmunity. And that is a pathway by which some individuals do develop autoimmune disease. Not really something we worry about in cattle, but certainly in other species and people, autoimmune disease can be a big problem. So again, there are sentinel cells of the immune response that are out in different tissues that are the first cells to see PAMPs or DAMPs. And these include dendritic cells, macrophages, mast cells, epithelial cells, and fibroblasts. You might be a little surprised to see mast cells on the list. Most of the time when we think about mast cells, we're just thinking about allergic reactions. But mast cells are found under the skin and under the GI mucosa and under the respiratory mucosa. And so, of course, those are all good places to identify an invading organism. And so it turns out that mast cells do have an important role as sentinel cells. And of course, these first three cells are all mobile cells that can travel around the body and migrate into regions of inflammation. Epithelial cells, of course, are on the surface of many the respiratory epithelium, the GI epithelium, of course, our cutaneous epithelium. So it's interesting to note that these are able to identify PAMPs and DAMPs, and again, makes sense because they're on the surface where the initial infection is likely to occur. A little bit about macrophages because they're so important in activating inflammation. They can identify and kill pathogens via many pathogen recognition receptors. They activate, or they're able to get rid of pathogens by phagocytosis. They also secrete antimicrobial products. They produce many molecular mediators of inflammation, such as prostaglandins and pro-inflammatory cytokines like IL-1, TNF-alpha, IL-6. I know that the cytokines get to be alphabet soup pretty quickly. If you can remember just a handful of them, it can be helpful when you hear different presentations or maybe get literature about new products from companies. And so IL-1 and TNF-alpha, if those can stick in your mind, those are good to keep in mind as inducers of inflammation. And they, of course, do that very effectively. And again, inflammation, while it is bad if we have too much of it, it's important to have some inflammation because it is what activates the immune response. Just a few words now about prostaglandins and other lipid mediators of inflammation. So recall something we all get taught, I think, in probably physiology or pharmacology in vet school. All cells have membranes, and cell membranes can be acted on by an enzyme called phospholipase A, leading to release of arachidonic acid. And then arachidonic acid can be acted on by cyclooxygenase, leading to production of prostaglandins, thromboxanes, and prostacyclins. And these are all associated with inducing inflammation by increasing blood flow, vascular permeability, and inflammatory cell influx. And you'll remember that there is cyclooxygenase type 1 and also type 2, and we'll talk more about these in a little bit. When we talk about inflammation, again, we also talk about cytokines. We've already mentioned tumor necrosis factor alpha, IL-1, IL-6 as important pro-inflammatory cytokines. There are really hundreds of other cytokines as well. You can think of cytokines as essentially chemical messages that one cell sends to other cells to influence them. Cytokines then do pretty much everything a cell does. It does at least in part through release of cytokines. So there are cytokines that activate the immune response, cytokines that direct specific types of responses, be they antiviral, antibacterial, or antiparasitic. And cytokines, while necessary for immunity and for inflammation, they can, if produced in excess, lead to death through septic shock. So I mentioned earlier that sentinel cells identify PAMPs and DAMPs through their pathogen recognition receptors. These pathogen recognition receptors are found pretty much anywhere a cell might find a pathogen, and that includes outside of itself. So there are pathogen recognition receptors on the surface of cells, or the cell might engulf something that turns out to be a pathogen. So it turns out that they also have pathogen recognition receptors inside endosomes inside of cells, and even just inside the cell cytoplasm, because some pathogens like viruses and certain bacteria can rapidly get into the cytoplasm. And so here's a little schematic demonstrating the sites where cellular pathogen recognition receptors can bind to PAMPs or also DAMPs. And so some of them occur on the cell surface. There are different toll-like receptors that occur on the surface of cells. Inside endosomes, toll-like receptor 3, 7, and 9 are found. And then some of the pathogen recognition receptors in the cytoplasm include RIG-I, or the NOD-like receptors, or MDA5. And so all of these different pathogen recognition receptors can bind to different types of PAMPs. The binding of a pathogen recognition receptor by a PAMP or a DAMP initiates a signal transduction sequence in the cell. So this is essentially a chain reaction of molecular events that causes the cell then to do things, either to produce cytokines that activate the inflammatory immune response, or it might cause the cell to start to replicate or produce other sort of mediators. The mixture of cytokines produced by a cell will determine the kind of immune response. And so if it's a certain set of cytokines, it'll be an antiviral response. Other cytokines will lead to antibacterial or antiparasitic responses. So here's a little schematic of how this all happens. In this case, demonstrating the consequences of a PAMP binding to a pathogen recognition receptor, using the example of the PAMP that's LPS or endotoxin, binding to its receptor, toll-like receptor 4. So here we have a macrophage with lipopolysaccharide represented by the green spiky structure, binding to the red rectangle, which represents the toll-like receptor 4. And here we've magnified it so you can see a little bit better that LPS binds to TLR4, and the black line there represents the surface of the macrophage. Well, right inside the macrophage's cytoplasm, there's a molecule called MyD88 that's associated with TLR4. And once LPS binds to TLR4, then that's going to lead to multiple different intermediate molecules being activated. And then these are going to lead to activation of transcription factors in the nucleus that then cause DNA DNA transcription to occur, so that messenger RNA for different pro-inflammatory cytokines like TNF-alpha, IL-1 beta, and IL-6 to be produced, as well as cytokines like interferon beta. And so this I've demonstrated with one molecule, but of course the macrophage is going to have dozens or maybe even hundreds of these molecules, so this is going to be a massively amplified reaction. Now, maybe you, like these poor cattle, are thinking, why does this all matter? Well, here's a picture that I'm sure looks very familiar to all of you. This is a gross image of fibrinous bronchopneumonia, such as we see due to Mannheimia hemolytica. And so Mannheimia hemolytica is a gram-negative bacteria, and so I've tried to represent that it's got a capsule. That's what this sort of lighter outer color is here. It kind of looks like hot dogs and buns, but it's really a schematic of a gram-negative bacteria with a capsule. And so because it's gram-negative, Mannheimia hemolytica does produce LPS. And as we just discussed, and we'll represent LPS as the little red cylinders here, it also produces a molecule called leukotoxin. And LPS, as we've discussed, will associate with toll-like receptor 4, which has a partner molecule called CD14 on the surface of macrophages. And when that happens, the macrophage, that chain reaction of events we just talked about, is going to lead to production of TNF-alpha, IL-1 beta, also prostaglandins. And that's going to lead to activation of endothelial cells in small blood vessels in the area. And that's going to lead the blood vessels to become leaky. And that's going to lead to edema, hemorrhage, and thrombosis. The activated macrophage is also going to produce interleukin-8, which is going to induce neutrophils to come in. It's a very potent, what we call chemotactic factor. It calls in, it's a chemical substance calling neutrophils to migrate in. And of course, the neutrophils are coming in to fight off the Mannheimia hemolytica. But in one of life's dirty tricks, to leave the blood vessel and get out into the tissue, the neutrophils have to upregulate expression of this pair of molecules called CD18, CD11A, or sometimes known as LFA1. And the important thing about that is that leukotoxin is produced by Mannheimia hemolytica, and it specifically binds to CD18 on ruminant leukocytes, leading them to die. And so when the neutrophil is just doing its job, it increases expression of LFA1 so it can leave the blood vessels and get out into the tissue. What's going to happen if the Mannheimia hemolytica is there? It's going to produce leukotoxin, which kills the neutrophil. And when the neutrophils die, if they undergo necrosis, which they sometimes do, then their contents leak out. And their contents include proteases, elastases, so these are enzymes that can break down tissue components, as well as reactive oxygen species that can cause lipid peroxidation and damage of membranes. And altogether, this is going to lead to tissue necrosis, finally resulting in fibrinous necrotizing bronchopneumonia, which we saw in the previous picture. So this is how these sort of PAMPs associating with their receptors can lead to inflammation and other processes that culminate in the BRD that we all know so well. And the relevance of this has been shown by research that showed that temporary depletion of neutrophils from cattle prior to experimental Mannheimia hemolytica infection significantly decreased clinical signs of disease and lung pathology. And this was shown in a couple of different studies. This then demonstrated the central role of inflammation mediated by neutrophils in disease due to Mannheimia hemolytica. Now, I want to hasten to say that this doesn't mean that neutrophils are all bad. The cattle that were infected with Mannheimia hemolytica in the absence of neutrophils weren't able to clear the bacteria, but in the short term, they had much less disease and much less lung pathology. So in the long term, it wouldn't have turned out too well for the cattle if they didn't have neutrophils, but in the short term, the neutrophils certainly did mediate the damage brought on by the Mannheimia hemolytica. And Mannheimia hemolytica isn't the only example where the inflammatory response is part of the problem. We see in bovine respiratory syncytial virus infection similar findings. BRSV infects epithelial cells of bronchi and alveoli. These infected epithelial cells release interferons and IL-8. The interferons activate natural killer cells and T lymphocytes to kill infected epithelial cells. And so that's necessary to get rid of the virus factories that these epithelial cells essentially become. However, that also, of course, leads to loss of the epithelial cells, which, when they're functioning normally, help to, through their mucociliary apparatus function, keep bacteria out of the lungs. The other thing that happens with BRSV is that, again, you have a lot of IL-8 being produced, and that causes neutrophils to come in. And we also see, and of course, the neutrophils can cause damage in the airways in BRSV, just like we described for Mannheimia hemolytica. Now, it's a little bit different in degree. We don't tend to see the necrosis and the thrombosis that we see with Mannheimia, but we certainly see a lot of neutrophils in the airways. In BRSV, we also see activation of lung mast cells, probably in part through their role as sentinel cells. And they can lead to release of histamine, which causes bronchoconstriction. And so the net effect is bronchiolitis, alveolitis, and respiratory distress, such as seen in this calf here. And so this is a photomicrograph of lung from a calf that was infected with BRSV. This is an immunohistochemistry image. So the brown staining is where the virus is. And you can see there's lots of virus. And what's really very, I think, amazing is to see this airway. So it's almost like a brown ring, right? Well, basically, those are the airway epithelial cells, and they are all infected with BRSV, which you can see by the brown staining. And then the airway is completely filled with neutrophils that have come in response to the IL-8. And so, of course, if you've got an airway completely filled with neutrophils, you're not going to be able to breathe very well. And you can see a lot of the virus out here in the alveolar epithelial cells, and a lot of just, I mean, really remember when you look at a lung under the microscope, it should be mostly air, and here there is really hardly any air. And so, yeah, the not only are neutrophils coming in response to the virus, but lymphocytes as well. So infection with viruses and bacteria that contribute to BRD induce inflammation. Inflammation is necessary to activate immunity, but in excess, inflammation contributes to morbidity and can even lead to death. So given all this, it is logical to consider treating BRD with anti-inflammatory drugs. And both corticosteroids and also non-steroidal anti-inflammatory drugs have been used to treat BRD. Just going to say a few words about corticosteroids. We don't have, well, just first about corticosteroids. What do? They inhibit inflammation through multiple mechanisms. They decrease expression of COX-2, which is the enzyme that is necessary to produce prostaglandins. Corticosteroids decrease expression of pro-inflammatory cytokines. They increase expression of annexin-1, which inhibits phospholipase A, which you'll recall we said breaks down cell membranes to form the arachidonic acid, so that's going to further decrease prostaglandin production. And they decrease movement of neutrophils out of blood vessels. So basically, corticosteroids do lots of different things that counteract the inflammatory response. But there haven't been very many studies looking at corticosteroids in BRD, and maybe it's just because people started using them and the word got out that they didn't work very well. So there isn't really very much published, but here's a trial that does demonstrate the fact that corticosteroids even though it sounds like they ought to be good, are not very good when it comes to BRD. So in an experimental, well, let me just back up. Before I get into the clinical trial, what I want to describe first is this experimental challenge study. So in an experimental challenge study of calves exposed to Mannheimia hemolytica, corticosteroids did look good. So in this study, calves were treated with dexamethasone before and during experimental Mannheimia hemolytica infection. They got dexamethasone 6 hours before and immediately before at 2 mg per kg. That's a pretty high dose. And then every 12 hours, 1 mg per kg after infection. And the calves that got dexamethasone after they were before and after Mannheimia hemolytica infection had significantly less severe disease. And when they were necropsied at 48 hours after challenge, they had significantly less lung pathology. The calves that got dexamethasone had 6% abnormal lung versus the calves that got no dexamethasone had 69% abnormal lung. So that's a big difference. And so here is some evidence that suggests that, wow, steroids should be great for BRD. But, and this is what I was saying too soon, was that in a large clinical trial, it didn't actually work that way. This is an older study. As I said, we don't have too much new. Actually, I can't find really anything new in terms of steroids and BRD, but in this trial from a few decades ago, over 2,000 yearling cattle with BRD got either tetracycline, pyrilamine, which is an antihistamine, and or dexamethasone. So about half the cattle got dexamethasone once a day for 3 days, and about half the cattle got a placebo. And so what these investigators in this feedlot trial found that the percent of cattle that died was 6.9% if they got dex, 5.7% if they didn't. So no significant effect on death. But the percent of cattle requiring additional treatment, i.e. relapsing, was 23.8% in the dexamethasone group, 18% in the placebo group. And that was significantly worse for the dexamethasone group. So the conclusion of these researchers was that cattle treated with dexamethasone for 3 days after BRD diagnosis were more likely to require subsequent additional treatment. So, as I said, little additional published research evaluating steroids in infectious BRD in the field. We do use corticosteroids for individual cattle with severe acute life-threatening respiratory distress, such as fog fever or anaphylaxis or tracheal edema. And this makes sense. Certainly, I've done it myself. While it's logical, these are not evidence-based. So that's just to say that we don't have any controlled studies proving that this helps. So that's pretty much all I have to offer you for corticosteroids. So apparently beneficial in experimental challenge study with Mannheimia hemolytica, not beneficial in an older clinical trial, and not much published since then. So now let's get on to talk about non-steroidals a bit. So remember this pathway we talked about earlier, phospholipase A acting on cell membranes leading to production of arachidonic acid, then COX-1 and COX-2 acting on the arachidonic acid to lead to production of prostaglandins, thromboxanes, and prostacyclins. Remember that non-steroidals inhibit the function of cyclooxygenase. They may be non-selective and so inhibit both COX-1 and also COX-2. COX-1 is the constitutive that's associated with normal health of GI and kidney, the GI mucosa and the kidney. And so non-selective non-steroidals are going to cause toxicity through the effects on COX-1. There are also selective non-steroidals that have more specific effect just on COX-2, and those should lead to less inflammation with less toxicity. The non-steroidals commonly given to cattle with BRD include flunixin-meglumine, banamine, which is licensed for treatment of fever associated with BRD in the U.S. Meloxicam is licensed in some countries for BRD, but not in the United States. And then, of course, we have Florfenicol in combination with flunixin-meglumine, which is licensed in the United States for BRD. So, want to now just describe a couple of clinical trials looking at non-steroidals in different kinds of BRD. So the first I'm going to talk about is a study that looked at Florfenicol with flunixin in veal calves with BRD. This was what's called a non-inferiority trial, where they basically want to show that one is no better and no worse than the other. And that may sound like a funny thing to specify, and you may think, well, isn't that what we always want to study? But actually, from the perspective of analyzing the data, if you want to show non-inferiority, you actually usually need larger numbers of animals than if you just want to show that one is better than the other. This was done in 9 veal operations in Europe, calves purchased from local auction markets, no vaccinations, antimicrobials, or anti-inflammatories given at arrival. Then once they developed BRD, they were randomly assigned to treatment, and they were examined for 10 days, and evaluators did not know what the treatment was that the calves had received. So 106 calves that developed BRD were treated with Florfenical-flunixin, and their weighted enrollment was 52 plus or minus about 10 kilograms. 104 were treated with Florfenicol alone. So here we are essentially trying to tell whether or not there is a similar effect with Florfenical-flunixin versus Florfenicol. And what they found was that there was a decrease in rectal temperature for both groups, but the temperature went down faster for the group that got the flunixin, not really surprising. By day 3, there was no difference, but it did go down faster when they got the flunixin. So it was non-inferior for the number of calves with treatment excess on day 4 or day 10 post-treatment. So you can see here that the number of treatment successes on day 4 was 93.4% for the combination, 90% for Florfenicol, and the number of successes on day 10 was 67% for the combination, 60% for the Florfenicol. So there was no significant difference in performance and health outcomes, but it was better, the combination, for the decrease in rectal temperature. In this comparison of Florfenicol and Florfenical-flunixin, flunixin did not apparently improve outcomes. Now here's a description of Florfenical-flunixin in feedlot BRD. Now in this case, we're not going to really be able to compare the role of flunixin by itself because that wasn't the study design. What we can do, though, is look at 3 different antimicrobials for BRD treatment. So Florfenical-flunixin versus telanthromycin versus ceftiofur crystalline free acid. So these were auction market derived cattle. They got a dose of long-acting oxytet at arrival and other standard processing. And then when they were diagnosed with BRD, and in this study it was referred to as undifferentiated fever, meaning that they had fever with no other specific signs, they were then transported to a research feedlot and randomly allocated to treatment. The things that they looked at were first, second, and third relapse rate, BRD mortality, overall mortality, average daily gain, dry matter intake, slaughter weight, dressing percentage, carcass yield and quality, and financial returns. So this was a very thorough study evaluating the effect of these different treatments for feedlot BRD. So in terms of first BRD relapse, so needing a second treatment for BRD after being treated, cattle given the Florfenical-flunixin had a 3.5 times greater risk than cattle given telanthromycin. So they were more likely to require a second treatment compared to telanthromycin. However, for overall mortality, cattle given telanthromycin had a 5 times greater risk than cattle given the Florfenical-flunixin, and that was significant. Cattle given ceftiofur had a 10 times greater risk than cattle given Florfenical-flunixin. Unlike an earlier meloxicam trial, and I didn't get into this here since meloxicam is not approved in the United States, there was no effect on average daily gain or lung lesions. So meloxicam did look beneficial, but that doesn't help us since it's not approved. And here's the take-home message in this trial, which was actually done in Canada. The total economic advantage for Florfenical-flunixin per treated animal was $46 Canadian as compared to telanthromycin and $108 Canadian as compared to ceftiofur. So in this really well-designed, thorough feedlot trial, giving Florfenicol with flunixin while having increased risk for needing a second treatment did decrease mortality and then overall decreased expense. Now, we're talking about anti-inflammatories here, so I mentioned that trial because it included an anti-inflammatory, but of course we didn't have a group that had florfenicol alone. It would have been nice to do that because that would have given us more information about the benefits of the flunixin. So we can't tell in this trial if the positive effects of the combination were due to the florfenicol, the flunixin, or both together. I just want to say a few words about something new. So while we've looked at this pathway a couple times in this talk, where we remember that phospholipase A acting on cell membranes leads to arachidonic acid, then COX-1 or 2 acting on arachidonic acid leads to prostaglandins and so forth, there is another enzyme called 15-lipoxygenase, which can also act on arachidonic acid, and it leads to production of lipid molecules called lipoxins. And the interesting thing about the lipoxins is that they actually decrease inflammation. And so this is probably, the host, no doubt, evolved this pathway to try to limit the inflammation induced when we have activation of this pathway that we're looking at. And so while we activate inflammation, we also activate anti-inflammatory pathways to try to temper or modulate inflammation. Lipoxins and other related molecules are known as specific pro-resolving mediators, or SPMs. And so these are lipid molecules that suppress inflammation. So they're kind of the counterbalance to prostaglandins and leukotrienes, which are lipid molecules that activate inflammation. And production of SPMs is activated near the beginning of the inflammatory response, presumably to limit tissue damage that could occur if inflammation became out of control. Lipoxins and other SPMs have been associated with decreased mortality in mouse models of sepsis, so the mice that make more lipoxins are less likely to die. They've also been associated with resolution of airway inflammation in mouse models of asthma, and lipoxins have been linked to decreased asthma severity in humans. So interestingly, in a small study in high-risk beef cattle, cattle that resisted BRD, so they came in at risk for BRD, but never were treated for BRD, had significantly increased activation of SPM pathways at arrival compared to cattle that went on to develop BRD. So this suggested that resistance to BRD may be due to improved ability to limit inflammation. However, this is just a small study, and more work is ongoing, and we may have more interesting information regarding this soon. Actually, you're going to hear more about it, or actually you did already hear about it when Matt Scott, who I think spoke to you last night, talked a little bit about this work. So we are looking at SPM pathways being produced in high-risk stocker cattle at arrival, and have some evidence that it can help them protect them against BRD, and that may be because of limiting inflammation. So to summarize, infection and tissue injury both activate inflammation, and this is induced by PAMPs and DABs interacting with immune cells. Inflammation is necessary to induce immunity, but too much inflammation can be lethal, and inflammation does cause many of the lesions we know to be associated with BRD. Treatment of BRD with nonsteroidals decreases morbidity and improves growth in some cases, but the effects have been somewhat inconsistent. This might be because they aren't always given at the right time, or we may not have a complete understanding of benefits of harm, the benefits versus the harms of lung inflammation in BRD. We've got a little bit of preliminary evidence that endogenous lipoxins and specific pro-resolving mediators may have beneficial effect to mitigate BRD, so this may be the beginning of some new paths to therapy or prevention. I would say that we definitely need a better understanding of the role of inflammation in BRD. We have a little bit of information from some well-designed research, but there's a lot we still don't understand. And I'll be happy to take any questions.
Hey, Dr. Woolums, how are you tonight?
Hello, Harold. Nice to see you. Very well, thanks.
We do have one question in the chat box. It's dex dose in the large trial is quite a bit lower than the 2 milligrams per kilogram in the experimental trial? Would that make a difference in the outcome, do you think?
Well, sure. I think that's a great question, and it may. I defer to a lot of you folks on Dex because I usually try to come and talk about what I can find in the published literature and just help you interpret it. I think Dex has probably been tried a lot more than is described in the literature. And so my impression from what feedlot consultants and stocker consultants have told me is that it just really doesn't help if you use it too often. So somebody has said that. So there you go. But yeah, I think in terms of comparing those two studies side by side, there are a lot of differences, and one certainly would be the dose that the cattle got. I think mainly I just want to put that in there because, when you talk about the role of inflammation in BRD, it's very logical to ask, well, what about trying to modulate it? And steroids, of course, are very effective anti-inflammatories, and we even have some evidence that in certain experimental, tightly controlled settings, they really help. But they just, have not really panned out in the field.
So I want to ask a question about hydration. Does hydration have any effect on the inflammatory process?
Well, it's a good question because some of the cattle certainly come in looking dehydrated or probably are dehydrated, and blood flow is going to have an influence as well. I haven't really seen anything directly measuring hydration by any sort of controlled, repeatable method. I know Dr. Richardson's done some work on this, and I'm sure you know that too, Harold, looking at treating calves with fluids at arrival, and I think didn't really find a big positive effect, but that's not really answering the same question. So, all these inflammatory mediators are carried in blood and tissue fluids, and if the animals don't have adequate blood flow, I'm sure there could be an effect. But I will say it may not always be straightforward, right? You may think, oh, better hydration means better responses. But sometimes we have animals that come in looking like they're going to die, but they're kind of holding on, and then we get them reperfused and then they die. So I think it's because once you get the blood flowing around and those inflammatory mediators getting to all the organs where they can have negative effects in excess, that may be why we haven't always seen maybe why Dr. Richardson didn't see a clear linear relationship between giving fluids and getting better.
Any other questions for Dr. Woolums?
Yeah, Amelia, we've gone round the robin, and I think we've talked about it before at this meeting about other infections as they modulate the inflammatory process, and one of them are parasites. And can you summarize that for us as far as the role that parasitism, and I'm thinking about GI parasites, play in affecting the inflammatory system?
Most of what I'm the most familiar with is about the role of parasites in modulating the adaptive immune response. So if you've got nematode parasites on board, that's going to drive T helper type 2 cells. And we didn't really talk about those tonight too much, but T helper type 2 cells are important for making B cells make IgE. If you have worms, you want IgE because IgE is very important for clearing worms. But what's been shown, not so much in cattle, but in other species, is that if an individual, and in people, actually this has been shown in people, studies of people in places with certain parasites, if they've got a burden of some kind of parasite that drives a T helper 2 response, then they don't have the kind of T helper response that you need to clear intracellular pathogens like viruses or mycobacteria. That's the T helper 1 response. And the T helper 2 and the T helper 1 responses are sort of mutually exclusive. And so, not completely, but a little bit. And so there are definitely studies showing that in certain species, including humans, if you've got worms or some other kind of parasite that drives a strong T helper 2 response, then you will not have as good of response to something like a virus or intracellular pathogen. And so then, of course, that begs the question, if we have stockers come in wormy, And so they've all been driven to induce T helper 2 responses. Are they able to respond to viruses? Are they able to respond to viral vaccines? And we've done a little bit of work on that, and it wasn't quite as clear cut as I hoped it would be in the little study that we did. Other people have looked at it, but not really very thoroughly. So I think there's probably something there, but it's a little bit hard to identify anything very clear cut. Yeah, somebody says, I think I remember some old data showing a beneficial effect of vitamin C. Yeah, any comment? I've seen some of that data too. And, you know, I wish more stuff were published. When I got ready to do this talk, I thought there would be a lot more on Banamine out there, but I couldn't find very much. What I mean by that is controlled field trials like the one I showed you. There are other studies that I didn't get into where they challenge with Mannheimia and then they give Banamine or not, and, you know, the fever goes down and the calves look a little brighter, but so vitamin C, I know some consultants use vitamin C. I know others that don't. Some of these are big consulting practices where I know they do research and I assume they have data supporting the value of vitamin C. So that's what I always think about vitamin C. Some of the people who I know have the wherewithal to do the research use it, but I haven't seen the published work myself.
Any more comments or questions for Dr. Woolums on this subject?
I'll just say real quick that I was also surprised by the high dose of Dex in that experimental trial, and I'm not sure why they used that high of a dose. You know, I would never, I don't know, I wouldn't usually use a dose that high. So back when I was getting out of my residency, people sometimes would do that for horses with neurologic trauma, but that's even kind of fallen out of favor. So